Okay, I think we're live, so I'm going to go ahead and see what questions exist, if any. If not, I will be around for the next hour to address any questions you have about chapters 14 and 15. Okay, so I have a question. Does low map units always indicate linked genes? So let me think about that. Low map units, so linked genes is just a general term that we use to describe genes that are on the same chromosome and therefore don't experience independent assortment. So the degree to which they are linked is determined by how close they are located together on the same chromosome. So for an example, I can have a maternal chromosome and a paternal chromosome. And let's say we're talking about something like body color for a fruit fly, body color and, I don't know, eye color. And let's just pretend that these are the alleles for body color are on the same chromosome and eye color are on the same chromosome. So an organism who is little a would have a black body color, and an organism who is little b would have a green eye color. I don't know. So if an organism is heterozygous, big A, big B, little b, they would have wild type. Let's just say the wild type is orange body color and red eyes. If the alleles were located very, very close to each other, then in this case, we would see that the alleles are almost probably completely linked. In other words, we would see very, very few offspring that show recombinant phenotypes. On the other hand, if they're a little bit more spread out, like, I don't know, maybe here and here, in this situation, we might see, I don't know, 15, 20% of the offspring show the recombinant phenotype. But again, remember, if you ever see offspring that are not 50-50, which means that 50% show the parental, the other 50% show the recombinant. Anytime you don't see 50-50, that indicates that the genes are linked. Now, how linked they are just depends on how close they are together on that chromosome. Okay, so I have another question. When do you see a 9:3:1 ratio? You see a 9:3:1 ratio. Let me get my screen share going. Sorry. You see a 9:3:1 ratio anytime you have, number one, we have to be doing a dihybrid cross, so that's one rule. And number two, the parents have to be heterozygous for the trait. Both parents have to be heterozygous. And then finally, number three, in order to see a 9:3:1 or an approximately 9:3:1 ratio, we have to see, or the alleles, the genes, must be on different chromosomes. So in other words, the law of independent assortment must be true in order to see the 9:3:1. And the reason for that is that if the alleles do sort independently, when this male's germ cells go to make sperm cells, the male's chromosomes can either line up like this, or the male's chromosomes can line up like this. And so depending on how these homologous pairs line up during the formation of sperm cells will determine what allele combinations the gametes carry. So if the homologous pairs line up like this, we get the production of sperm cells that look like that. If the homologous pairs line up like this, which is equally likely, we get sperm cells that end up looking like, whoops, looking like this. So if we look at the entire population of this male's sperm cells that are produced when his germ cells constantly undergo meiosis, if we were to sample those gametes, we would see that approximately 25% of all of his gametes carry both dominants, 25% of all of his gametes carry both recessives, 25% would be carrying the dominant A, the recessive B, and the remaining 25% would carry the recessive A dominant B. So when reproduction happens, and again, the exact same thing is happening over in the female because they have the same genotype. She's just making eggs. When fertilization happens, what you end up with is, and assuming that this, well, obviously this fertilization is going to be random, each gamete is equally likely to do the fertilization. You know, this gamete doesn't have some sort of edge on the other gametes. When we do fertilization, we see that if we were to construct a Punnett square, which you probably should try to get away from doing Punnett squares just because they're time consuming and they don't really work for a tri-hybrid cross. They do work, it just takes an enormous amount of time to do them. If we construct a Punnett square, we can see all of the possible outcomes due to fertilization. And so if we were to make these crosses, like for example, if we were to simulate this fertilization, and I did that 15 more times, you know, I did it for here and here, I would get big A, little b, little b. So if I did all of those simulated fertilization events, what I would end up seeing is that 9 of the offspring would show dominant for both. This blank just means that it could be either a big B or a little b. It really doesn't matter. They're showing the dominant phenotype for B because they already have one big B. 3 would show dominant for one, recessive for the other. 3 would show recessive for one and dominant for the other. And then 1 would show recessive for both. And again, this is only if both parents are heterozygous and the genes are located on different chromosomes, which means that we see independent assortment happening, which means that we see these gametes being produced in equal ratios. Let me see if I can share this with you. Wait a second. Okay, I don't know if you can see this or not. This is something that I made last year that I just forgot to show you guys this year. This is just an Excel simulation of a mother with the heterozygous genotype and a father with a heterozygous genotype. And this is a dihybrid cross. And this is simulating fertilization. It's basically a massive Punnett square where we make about 700 offspring. And when you run the simulation, you see that the red bars represent the expected 9:3:1 ratio. So every time I run this simulation, I see that the number of offspring produced approximately fit that 9:3:1 ratio. And I think in the background, I even have a chi-square test running every time. So you can see that I think when the box appears yellow, we end up getting some crazy deviations that would cause us to reject the null. But notice how when I've pressed this simulation and I've ran it like probably close, there we go, we finally got a yellow. But notice that the observed offspring are, the vast majority of the time, they're fitting very closely to that 9:3:1 ratio. Every now and then it'll deviate due to chance. This is just showing that that's indeed what you see, that 9:3:1 ratio, if you take two parents that are heterozygous and the traits sort independently and you cross them. All right, I'm waiting for another question, so I'll be right back. Okay, so I see a question about the Hershey and Chase experiment, and just since I'm not getting any other questions about chapter 14 and 15, I can talk about that. So the Hershey and Chase experiment was an experiment conducted in the 20th century. Give me 1 second. Okay, sorry. So it was conducted in the 20th century, and it was looking at how viruses transmit genetic information into the host cell. At the time, it was thought, or it was argued by many members of the scientific community, that proteins were the molecule that was responsible for the transmittance of traits from one generation to the next or from one organism to another, because proteins are just so much more complicated than DNA and complex. So it just made sense to them that something so complex surely must be responsible for the complexity of life. And so what this experiment did is these little pill-shaped things are bacteria. What this experiment did is it took radioactive sulfur and radioactive phosphorus. Now, phosphorus is an element that we find in DNA, but not in proteins. Sulfur is an element that we find in certain amino acids, which make up proteins, and we don't find any of it in DNA. So let's make them, I guess, red. What we're going to do is we're going to make in one population, we're going to make the virus's protein radioactive. And so we'll make it glow red because we're using radioactive sulfur in this population, and their DNA is not going to be radioactively labeled. And then in the other population of bacteriophages, we're going to do the exact opposite. We're going to make it so that their proteins aren't radioactive, and we're going to make their genetic information radioactive because we're using phosphorus, an isotope, a radioactive isotope of phosphorus. And now we're going to let the bacteriophages do their thing and infect the bacteria. And the idea is that after the infection, that's terrible, after the infection, we're going to look at where the radioactivity goes. We're going to look at, basically, we're going to look at under which scenario, scenario 1 or scenario 2, do the bacteria become radioactive? Because wherever the bacteria become radioactive, that indicates that it's gotten that molecule from the virus. So in scenario number 1, what they found is that when they let the bacteriophages infect the bacteria, they saw radioactivity in the fluid above the bacteria. So in other words, they let the bacteriophages infect, and then they shook the vial or the beaker around, and to get all of the viruses to fall off of the bacteria. And then when they looked at where was the radioactivity, they saw that the radioactivity was just in the virus particles that were suspended and floating above the bacteria. But in this scenario, what they found is that after the bacteriophages infected and they shook up the beaker to get the bacteriophages to fall off, they found that the bacteria on the bottom were suddenly now radioactive, and that the solution on top of the bacteria had no radioactivity in it. So that told them that the viruses must be injecting nucleic acids into the bacteria, not proteins, because only when the virus's nucleic acids were radioactively labeled did we see radioactivity in the bacteria population after they were infected. So that experiment showed that, at least for viruses use nucleic acids as a way to transmit genetic information from one organism to another, or from itself to another organism. There's a question, why does recombination between linked genes still occur? That is thanks to crossing over. So let's take in an organism's germ cells, so these are going to be the gamete-producing cells. This is the maternal chromosome, this is the paternal chromosome. And let's say that this is a heterozygous organism, and the alleles are arranged in such a way that the dominant alleles are on the same chromosome for these traits, and the recessive alleles are on the same chromosome. So if we mated this organism with a homozygous recessive, the only gamete that this organism can make, the only gamete that this organism can make is one that looks like this, right? So if crossing over never existed, if there was no such thing as crossing over, then exactly half of this organism's gametes would be big A, big B, and the other half would be little a, little b. But that's not what we see. We see that there is sometimes this organism is producing gametes that are little a, big B, and gametes that are big A, little b, and this is because crossing over. Crossing over is a mechanism that has been favored by natural selection simply because it produces new allele combinations. And these new allele combinations could potentially benefit the offspring and make it more likely to survive and reproduce in an environment that's constantly changing. So in order to get these new allele combinations, we just need crossing over to happen anywhere in between the space between the genes. So if crossing over happens down here, that's great. But as far as these traits go, it doesn't produce any new allele combinations. So crossing over is going to happen between the loci, or the location, of these two genes. So if there's a crossover event, for example, between here and here, what we would get from that is something, something like this. Okay, now if that happens, now the allele combination that the gametes possess look like this. And we haven't produced any new genes here. We haven't made any new alleles. All we've done is we've changed the allele combination. And so if these gametes are produced, we wind up with organisms that show recombinant phenotypes, which may be beneficial for the organism. It may not have any impact on the organism's survival, or it may actually put the organism at a disadvantage. But that's the whole point of sexual selection. It's rolling the genetic dice. Oh no, I didn't share my screen. Crap. Oh, that's terrible. Hang on. It's in the notes. It's in the notes. This is what I was drawing the whole time. Sorry, I'm always making some sort of mistake. I'll get better every time. Even though you missed that little drawing as I was doing my explaining, in the notes that I shared and posted on my website, there's a really good animation that narrates the whole crossing over recombination thing. So if you're struggling with understanding how crossing over works and how it produces new recombinant chromosomes, then definitely check out that animation. It'll probably help a lot. Since I don't see any more questions, I guess I can go back and do a little bit of explaining since you didn't see that original drawing. So what I was doing is I was drawing the paternal. All right, let's call that the maternal. And let's call this blue one the paternal. So this is the chromosome that you got from dad. The red one is the chromosome that you got from mom. And what I was drawing is the alleles. The dominants were all located on the same chromosome, and the recessives were located on the same chromosome. So I was saying under these conditions, the gametes that you produce always, if crossing over never happened, are always going to look like half are going to look like big A, big B. The other half are going to look like little a, little b, because that's what's going to happen when meiosis 1 and meiosis 2 happen. When these homologous pairs separate, and then when the sister chromatids separate, you're going to end up with half of the gametes are big A, big B. The other half are going to be little a, little b. That's not what we see. And then if you were to cross it with an organism that was this, little a, little b, which is always going to make the little a, little b gamete, then if crossing over never happened, then you're always going to have offspring that look like this parent if this fertilization occurs, or they're going to look like this parent if this fertilization occurs. But the reason that we see recombinant phenotypes produced in this scenario is because you have a crossover event that happens somewhere between these two genes. If a crossover event happens somewhere down here, that's still a crossover, but it doesn't unlink those genes, because the crossover has to occur in a region between those genes in order to get the alleles to flip chromosomes. So if a crossover happens between this spot on the homologous pairs, then what we would see is this. We would see a chunk of the paternal move over, and we would see a chunk of the maternal move over, and then I would need to erase that. And make it look like this. So under these conditions, we can now get new allele combinations inside of the gamete. And if these are used for fertilization, remember they're always going to be fertilizing a little a, little b if this is the genotype of the other parent. Then we end up with organisms that have different combinations of phenotypes. They don't have brand new phenotypes, they just have different combinations of the phenotypes that already existed, which may or may not help them survive and reproduce. Pleiotropy. So there's a question, what is pleiotropy? Pleiotropy and polygenic inheritance are some of the most common forms of inheritance in humans, and they're complicated, so we really don't touch up on them that much. But pleiotropy, or pleiotropic inheritance, an example of that is cystic fibrosis. So pleiotropic just means that there is a gene that when expressed, it has a wide range of effects on the phenotype of an organism. And that's different from what Mendel studied. Mendel studied, for example, a gene for flower color. And the gene that he was studying, he called them characters, but the gene that he was studying for flower color only influenced flower color. Be a gene that controls flower color, influences how wide the leaves are, and also influences how tall it grows. I don't know. It influences multiple phenotypes. That's what pleiotropy is. And that's sort of the opposite of polygenic. Polygenic is where many genes influence one phenotype. And so the classic example for that is skin color. So skin color is determined by many gene interactions, and then obviously also how those genes are how those genes are expressed which is influenced by the organism's environment. But polygenic just means that there are multiple genes that contribute to one phenotype. Can unlinked genes become linked through crossing over? No, because by definition, genes that are classified as unlinked, the reason that they're classified as unlinked is because they're on different chromosomes. So chromosomes don't participate in crossing over with non-homologous chromosomes, unless you're talking about an error. Sometimes they do that, and that's not a good thing. But if we're doing crossing over between these homologous pairs and these homologous pairs, if these genes are not linked, the reason they're not linked is because they're on completely different chromosomes. So there's no way that we can suddenly make them become linked unless these chromosomes did some crossing over, or we don't even call it crossing over at that point, we call it translocation, because it's not supposed to happen, so it has its own different unique name. But the opposite of that, let me go in a different direction with that question. Can genes that are on the same chromosome behave as though they were on different chromosomes? And the answer to that question is yes, if the genes are really far apart. So, for example, these genes are so far apart from each other that a crossover event between them is virtually guaranteed. And if a crossover event between two genes is virtually guaranteed because of how far apart they are from each other, then isn't that the same thing as saying that about 50% of the gametes produced are going to look like this, and about 50% of the gametes produced are going to look like this because of how frequently crossing over happens? And so if crossing over happens so frequently that you end up with an equal proportion of gametes, that's the same scenario you get when the genes are on different chromosomes. So anytime we do a heterozygous cross with a homozygous recessive, I guess a heterozygous, and we cross it with a homozygous recessive, whenever we see a 1-to-1 to 1-to-1, two things are possible. Either we're seeing this because the traits are located on different chromosomes, or if they are located on the same chromosome, they're so far apart from each other that they behave as though they're unlinked. The question, could Mendel have been some studies about linked genes like this? Yeah, I think what you're asking is, did Mendel study linked genes without knowing? Yeah, kind of. If you look at all of the traits that he studied, some of those traits, some of those alleles or genes are actually located on the same chromosome. But Mendel got really lucky because even though they were located on the same chromosome, they behaved as though they were located on different chromosomes. It wasn't until Thomas Morgan came along in the early 1900s that gene linkage was discovered and made known to the scientific community. So the 7 or 8 traits that Mendel chose to study, there was a little bit of luck in him selecting those traits because he ended up selecting traits that either sorted independently or behaved as though they sorted independently because they were so far apart on the same chromosome. But if he had chosen a trait, or if he had chosen to study traits that did not display independent assortment because they were on the same chromosome and they were really close together, then his conclusions may have been completely different. Or he may have been the one who discovered linked traits and not Thomas Morgan. I mean, we don't know. Yeah, if the alleles are, okay, so the question is, if the alleles are really far apart, will they have the same ratio? I think what you're asking is, if the alleles are located very far apart on the same chromosome, do we see the production of gametes with the same ratio of allele combinations? And the answer is yes. So if the alleles are located very far apart, then what we see is that approximately 25% of the gametes produced look like this, 25% of the gametes produced look like this, 25% look like this, and then 25% look like that, which is the exact same thing we see if we're studying traits that are located on different chromosomes. All right, so we got about 20 more minutes. I haven't gotten any more questions, so I'll be right back. All right, since I'm not getting a lot of questions, if you have anything that you want to cover on chapters or on chapter 16, feel free to ask. Is there anything else important that we haven't covered? I mean, for this unit, yeah, there's the rest of chapter 16 and then some biotechnology, and then some of chapter 17. Is there anything else important from chapter 14 and 15 that we haven't covered in this Google Hangout? Yeah, I mean, there's a lot of stuff that I could cover. But you need to ask, or I'm waiting for you to ask the questions on what you would like to focus on specifically. I mean, as far as chapter 14 goes, there's the basic Mendel inheritance, which assumes that you understand chapter 13, meiosis. I think a lot of people are still iffy on meiosis, and that's making 14 and 15 a real struggle. There's basic probability with genetics, but I've uploaded 2 genetics practice problem documents. So I would encourage you to go through that first before coming to see me for help, and that way we can focus on specific problems. These practice problems have you go through all of the Mendelian ratios, the 3-to-1, the 9-3-3-1, the 1-to-1 to 1-to-1, genetic linkage, sex linkage. So these are really good documents to use. There's pedigrees, which is studying the transmittance of traits throughout multiple generations. And then 15 is mostly just everything about non-Mendel inheritance. So there's a lot of examples of non-Mendel inheritance. I've spent the most time in this session talking about linkage. That is not the only form of non-Mendelian inheritance, though. But again, there is a PowerPoint sort of cheat sheet up on the website. There's chapter 15 itself, the guided reading, the practice quizzes and tests in the book and online. So just make sure that you're utilizing all of those resources, especially if you're discovering that you're having a really difficult time grasping 14 and 15. Uh-oh, I'm glitching out. I don't know what that means. Is my sound cutting out? Yep, I am being told that I'm glitching out. I don't know what that means though. Is my audio cutting out? Let me try something really quick. I don't know if I'm still having difficulty or if the broadcast is still chopping up, but there's about 10 more minutes left, and I'm gonna just wait on some questions. I try not to review the entire unit in these Hangout sessions because I don't want to end up just reteaching entire units in person and then again over the internet, but I will answer specific questions you have about the unit. All right, we've got about 5 minutes left. I haven't gotten any new questions, but I'll be here for 5 more minutes in case any questions pop up. All right, it's 8 o'clock, so I'm going to go ahead and close this down. This was a slow night. I'll probably hold one or two more of these before the exam, but the best way to utilize these sessions is to come prepared with questions, and that way we can maximize our use of the hour. But I will see you guys tomorrow. Have a good night.